So the data is available for the genome, and you go to potatogenome.net, and that takes you to Potato Genomics, which we support that site. And essentially, you could download all the data. It's freely available. You can do a blast search, and then you can look at it in a genome browser. So what I want to show you is just a few screen capture shots of the genome browser, just to make sure you're all familiar with this. So you enter the site, and you can select a region. You can enter in a text here. You could search for something like a kinase or starch, and this would generate a return for you. You can also type in scaffold with its coordinates, and then this is the overview window right here. So this looks like it's a 4 megabase scaffold, and we're looking at this position right here. And then we see a gene overview region right here that's highlighted in yellow. And if we look at that in more detail, you can see here that there's actually 4 annotated genes. This one's an adenosine 3-phosphate 5-phosphate transporter. And then there's a gene here, this vacuolar iron family transporter, and a set domain containing protein, and then a Kelded motif domain protein over here. So these are the loci. These are the loci. And then we have different gene models to represent those loci. And so what's shown here is the BGI representative gene model. So this is the model that is the longest coding sequence. And then we actually have isoforms that we have predicted based on the transcript evidence. So you can see in this case over here, there's only 1 isoform. In this far case here on the left, there's 4 isoforms, and there's quite a few isoforms over here for this gene right here. We've also aligned the predicted protein of these loci to UniRef to get any similarity to proteins in the public database. And we've also aligned them to the Sanger EST. So if you wanted to look at a longer EST sequence, you could do that. And then what we've placed is 2 different tracks of the SolCap Infinium data. We've placed our candidate Infinium SNPs. So these are the 69,000 very, very high confident SNPs that we detected from Snowden, Atlantic, and Premier. And then we also have only the SNPs that are on the SolCap Infinium array. So here we have 5 SNPs that are high confidence SNPs, and only 1 of these is actually on the array, because the array only was able to hold a fraction of what we actually were able to predict. So we also have alignments of the predicted proteins to different dicot genomes, Arabidopsis, grape, and poplar. So what's shown here are called what's called the wiggle tracks, and what these are is the transcript evidence for that. So what these are, each one of these is a different library. So this is tubers, leaves, callus, wounding, and they're really just a heat map, an intensity map of the expression that we detected using RNA-seq. So the darker the blue color means that there was more expression of that exon. So these are the exons of the gene, and if you stitch them together, you'll get the actual gene model. And so this is good to just do a quick look to see what tissues your gene is expressed in. It can also give you some information that maybe the gene model is not annotated properly. So other accesses, there's a blast search. It's supported with all the blast family. You could download everything. You could download the transcripts, and you could download the proteins. Okay? If you want to make your own genome browser, you can do that as well. We provide the GFF.